we'll talk about the polymorphism, but I think the main point of the paper, as I see it, is using this polymorphism to illustrate how this drug may have important advantages over rituximab. And one of the differences, and there are several differences, including the fact that this antibody is humanized as opposed to chimeric for rituximab. And we talked about the broader spectrum of B cells that are targeted. But one of the other unique aspects about this drug is that it's glycoengineered and afucosylated, and this enhances the affinity of this drug for the FC receptor. The affinity, this enhances the affinity of macrophages and natural killer cells for the FC portion of this antibody, which is bound to B cells and facilitates antibody-dependent cellular cytotoxicity. So this is all complicated, so I do have a figure that I can share with you that I think makes this point more clear. What we have here is the B cell, and we have the CD19 protein, and we actually have two antibodies that are going to be relevant to talk about. But first, let's talk about Inebilizumab, which is a monoclonal antibody. The other protein or the other antibody we can talk about is the protein or the antibody that is actually causing the damage, the anti-aquaporin 4 antibody. But we'll get to that afterwards. And here are the natural killer cells or macrophages, which express a protein, a receptor called CD16. And the polymorphism is of this CD16 protein. And as is the case with all single nucleotide polymorphisms, there are 3 genotypes. But probably the most important one to talk about is the FF, where there's 2 phenylalanines at this locus, this polymorphism locus, and the F alleles encode a receptor that has lower binding affinity for the antibody, whereas the valine polymorphisms, those who have a single or 2 copies of valine, have higher affinity. And one observation that had been made in the past from South Korea is that when using rituximab, patients who had the lower affinity allele that would result in less binding of this receptor, and accordingly the effector cells, the macrophages and natural killer cells to this antibody, had a less good response, presumably because they weren't binding to this antibody and carrying out the antibody-dependent cellular cytotoxicity. And the key observation in this study was that this polymorphism did not have an important difference on the efficacy, measured in a couple of different ways, the relapse rate and also the depletion of B cells. There was a slight nominal difference, but overall the results were not statistically significantly different. And, you know, to me, what this tells us is that this afucosylation, which was theoretically predicted to enhance the affinity, seems to be the case. And we're using the patients with this polymorphism to illustrate the fact that Inebilizumab, because of its afucosylation and increased binding affinity, essentially neutralizes the differences that were observed for rituximab in South Korea. Another observation that hadn't been looked at, but you know, when you have this polymorphism, the other thing that it might do is if you substitute the Inebilizumab with the aquaporin 4 antibody, And if you substitute in this figure the B cells with astrocytes, which are the target for aquaporin 4, theoretically, patients with the veiling polymorphism, which is higher affinity, may have more severe disease. It may actually increase the binding to the aquaporin 4 antibody and more target damage. And in fact, when they looked at the characteristics of the patients that were enrolled in this study, a subset of the patients who were included in the N-Momentum study, the attack rate was significantly higher in those who were veiling carriers compared to those who were homozygous for the phenylalanine allele. So, really verifies that this polymorphism does have functional effects. But the key focus in this study was in terms of how this might influence the efficacy of inebilizumab. So I think one of the key questions that often comes up, because we've been using rituximab for many years in patients with neuromyelitis optica spectrum disorder. We now have generic forms of rituximab. Rituximab is very much less expensive. And should we start patients on rituximab, which is not FDA approved, I might add, or should we use the FDA approved but more expensive agent? And similarly, when patients are on rituximab and experience on-treatment attacks, big question is, well, what do we switch to? We now have 3 FDA-approved agents. And what had often been said is, well, if a B-cell depleting drug is not working, perhaps there's no advantage switching to another B-cell depleting drug. I think this just draws differences between rituximab and inebilizumab and says, yes, there may be a number of rationales, including the broader spectrum of B cells, but also the afucosylation, increased binding affinity. And in fact, this seemed to be empirically supported by another study that has been published, where 7 patients who were enrolled in the N-Momentum study who had had prior attacks while on rituximab, none of them during the inebilizumab study experienced any on-treatment attacks. So there may be reasons why inebilizumab is superior to rituximab, both theoretical and actually demonstrated in this study.